from me. It's great to have you here, and I must say that actually right now after the talk about the cows, I feel it's very difficult to talk about plants also for me, and you must be exhausted having all these different topics. At least I started to think that we should try to develop mouse with greater milk production, something would be a line of research for the future maybe. I start with a slide which is from Australia. You see this kind of nice trees, which is called grass trees. It's a very interesting ecosystem in the world. And if you have opportunity to go there, I really recommend to go. And then this is like the heart of our studies. We are interested in general is what determines the vegetation distribution on Earth. And then we really would like to look at this from a broader perspective, rather than only focusing on Estonia. Because, you know, in big climate models, Estonia is only 1 pixel. We tend to think that it's very diverse, and it indeed is, but in reality we need to generalize if we want to understand things on the broader scale, and then we are really focusing on from forest to deserts. And then the idea, what we finally ultimately need to know is how plants acclimate to their environment, is how they can cope with all these different conditions. They cannot run away if it's too hot or too cold, they have to stay there. And in particular, we are doing a lot of work and to understand the effects of different abiotic and biotic stresses in plants' environment. And just to give a few facts about our team, when I came to this university, it was 2006. I was working actually in Italy at that time at University of Tartu and shared jobs, and then I got a professor position from Estonian Academy of Sciences and from this university, and I started from zero, without any equipment or anything. And now we have like quite a well-staffed team, which is relatively young for Estonian standards. And this is mainly the work that has been done here. And then we have had quite many projects in these days. So in general, we are interested really from broad perspective, and here's these red squares, actually places where we have been doing, or myself actually have went there and then done field work. And it's very important, I think, as some people work only from computers, but it's important to go there to fully appreciate how the nature looks like, especially like here is one spot in a tropical rainforest in Malaysia, and then you read the papers and then you get quite an understanding how the plants are working. But if you go there and then just to get the process of getting this data through the mud and mosquitoes and the danger of malaria and all these tropical diseases, so you can fully appreciate how difficult actually it is to do work in many spots in the world. I must also say that most of the plant science research is focusing on these plants, which is Arabidopsis. Basically is a weed. It grows many places. It grows also in Estonia, and it's annual weed. And it's amazing how much resources I actually spent to understand this plant. This was the first plant that was a full genome was sequenced, and this is a very nice model ecosystem, model plant system. But on the other hand, it's actually very limited because it doesn't really make wood, or it doesn't grow in dense stands. This plant grows when there is resources available. It's not particularly stress tolerant. If there is water, this plant grows. If there is temperature is high enough, this plant grows. But if there is stress, then this plant is completing its life cycle. So it's this kind of ephemeral life cycle, very fast grow when the resources are available. And some people say you can make a salad from this, and maybe you can grow a super Arabidopsis, but it's in reality it's very limited. Also, I must say it's very useful in a way that the sequence is available, and you can do transgenics very easily, and you can study a molecular mechanism of regulation of plant processes, but you cannot scale for Arabidopsis to the world. So this is why we are actually really collaborative and then try to integrate with many teams all over the world. We are interested in global change, and this is something which has been measured already for a long period of time, and this is a reality that CO2 concentration is increasing. And actually, you can see this change in the lifetime of scientists here in Estonia. Here is a very famous professor, Agulais, who was one of the first in photosynthesis research, discovering many things well in advance when it was done in Western world. So it was first initially publishing in Russian, and now it's publishing in English. So it's still around and has been doing a lot of work. And what you see here is that CO2 concentration tended to be 320. then my BSc supervisor, Professor Olevikun, when he was studying, doing his works, and it was 340. And when I was doing my BSc thesis, it was 360. And now it is important actually because photosynthesis is a process for which carbon is fixed from the atmosphere. This depends on CO2 concentration according to this hyperbola relationship. So it's increasing with increasing CO2 concentration. And in fact, we are living in a world where photosynthesis is increasing. And also when Agulais was doing his measurements, it was getting smaller numbers than Olevikul or I was getting. So in fact, if we look also back in the literature, we must also consider this, that CO2 has risen to interpret the data. And in fact, now actually in May, it was 400. And this is like this kind of magic thing. It was like a lot of public attention, not so much maybe in Estonia. People barely noticed it, but it's actually a really, really important thing. This was considered like a milestone. Now we are 400, and it keeps increasing. So it's like a really, really important thing. In fact, on the basis of this increase of CO2 current rise in CO2 concentration, some people are saying that this is a good thing because we are getting more carbon fixed. And in fact, this is true. Actually, plants are photosynthesizing now much faster than they did in the past. And in fact, in the United States, some scientists said it's actually like they published the papers of many benefits of rising CO2, that we should not actually do anything to really avoid increasing CO2. It's actually, there is a lot of skepticism in the United States, and many scientists also testified in the Congress telling that, no, we need actually keep rising CO2. It's good for the plants, good for society. But on the other hand, there is the effect of greenhouse effect, which maybe we think we fully understand it, but we are probably not really there to understand it. But it seems that temperature is increasing at much faster than in the future. So in the past. So this is like a really important issue with rising CO2, and this is why Europe is doing a lot with trying to reduce CO2 emissions. So that's important, and also United States starts to act in the future, hopefully more aggressively to combat these emissions and so on. So this is an important issue. And here's what is expected actually in different environments. This is a picture taken from Intergovernmental Panel for Climate Change Report 2007. Now a new report will appear in this autumn. And these are computer model prediction for 3 different scenarios. What you see here is that there is quite a bit of warming expected within the next 100 years in northern latitudes. And for Estonia, this is like an opportunity, but in many other places in the world, especially for the plant, it will mean a lot of stress if this will be the reality in the future. In fact, this is all based on computer models. And these are very complicated models, actually. And then you also see the large differences depending also on input scenarios and then many things. So this is something why the skeptics of climate say that this will not happen, because this is based on computer models. But this is the best understanding so far. Well, if we look how plants react to temperature, then here is again the photosynthesis rate, carbon fixation. It responds to temperature according to so-called bell-shaped curve, or a curve with an optimum. And actually on the basis of this curve, people were predicting that in the future there will be a big problem. Like in Amazon, the rainforest will become a desert in 20 years' time. And this was very bad science, I must say. And it was actually like, in news, it made actually big stories. It's just in the Independent, but it was in all major newspapers that there will be a major problem. Actually now, there should be a desert in Amazon. And it was not true, and because the models were so bad, actually. In fact, this was published in major journal, in Nature, but in fact it was, and why it was bad was that in these models they are assuming the same curve for tundra and for tropics. It's basically exactly the same, and like completely rigid curves, that, like a fundamental curve that no matter what, plants are performing according to this curve. But in fact, what plants are doing, when they are growing at a certain temperature, they are like having an optimum here, and when the temperature is rising, then the curve is shifting. And this is called acclimation. Adaptation and acclimation to the environment where they are growing. In fact, those plants growing here, And then there they have different temperature response curves. And also when future temperature is rising, then this will actually change a lot. And this is where the people are now working a lot. Actually, in none of the global models, this has yet been included. It's physiologists, we know this response very well, but it has not been really incorporated in these large-scale models, which is actually fundamentally changing our understanding how plant will respond to future conditions. And this is what we are trying to now also experimentally verify. And of course, there is probably what we are suggesting is evergreens are shifting and there will be changes in vegetation distribution, but not these sort of things that we are converting from tropical rainforest to desert just in one go. It will not happen. This is actually like really an example of bad science if people are saying this. So this is a topic, understanding plant responses to global changes, a topic of our Center of Excellence. Just probably you have seen this already. This is idea to catalyze collaboration amongst the teams, and also very important for Estonian relations to provide additional funding. And then the mainly funding comes from the European Commission, and also by participating institution is 5% is a self-contribution. And I'm in charge of Center of Excellence in Environmental Adaptation. We have 5 teams involved from 3 Estonian universities. So this is also quite rare that there is actually 3 institutions are involved. And there are right now altogether 12 Centers of Excellence. There were 2 calls in 2001 and in 2011. And our Center of Excellence ranked first in the second call in 2011. There were, by the way, 27 proposals. So it's actually very fierce competition, as in Estonia is in particularly difficult. Well, you know, this is another topic, and actually it's a major topic we are working right now in the context of global change, and this is understanding the role of volatiles emitted from plants. The point is that like worldwide, plants are the major emitters of volatile organics. Just to put this in perspective, from anthropogenic sources, from cars, from everywhere, basically there is 100 teragrams. So it's, 1 teragram is 1 million ton, so it's a very big number. And the plants are making, from human sources, 100 teragram per year, and from plants it's about 10 times or 11 times more. So these plants are really the major emitters of volatile organic. And also to put this in perspective, if you think about the annual carbon fixation for the planet Earth, it's about 5,000 teragram. And actually 5,000 teragram is fixed, and 1,000 teragrams, about 1,000 teragrams is released as volatile organics. And these are very active compounds. It's not like CO2 or something. We are dealing with very, very reactive compounds that are reacting in atmosphere very quickly. And you see also here is like isoprene is a major source. Almost half comes from isoprene, and there are also monoterpenes, the plant compounds. And why they are important is actually that these compounds are involved in forming, together with nitrogen oxides. Nitrogen oxides are mainly anthropogenic origin, come from combustion engines where cars are driving. It's very difficult to control actually. So this is like a major source here, and during a short light period when there is solar radiation, so blue light, there is this oxygen atom released from NO2, and then this together with deoxygen immediately forms ozone. And ozone is a major pollutant. It's actually very, very big problem. There are also at European level, and in particular in US, this line of research is actually much more developed in US, I must say. In Europe, we are actually especially this pyrogens emissions, they have been working on this field very actively and also included already in ozone control strategies from 80s. And then now in Europe, the understanding is also coming, but we are considering this like something we cannot do anything. Of course, we can change the species that are emitting and so on. Anyway, these reactive oxygen species are further forming secondary organic aerosols. And further, when they condense, they are forming cloud condensation nuclei. And this is actually the link to large-scale effects on climate. Of course, ozone itself is also a greenhouse gas, like CO2 is actually much more active greenhouse gas than CO2. But the major effect goes for organic aerosols, and here is something what you see actually when there is a period of organic aerosols. Also like this spring, for instance, in major airports, you come and you see this lila or blue haze in the atmosphere. That's organic aerosols, and mainly due to plant emissions. So that's a very important thing. And then the more aerosols there are, the less solar radiation penetrates. So it's actually working against the rise of CO2. Here's also in the clean air was a paper in Science by a Finnish team. So it shows that in clean air you have actually enrichment of air with aerosols. Here's another one about the boreal forest aerosols. And here was a paper in Science that if you clean the air, you potentially heat the planet. And that's right, actually. In the future, we are expecting much cleaner air because we are reducing the anthropogenic emissions. Of course, now it's also like due to strange ways, people start burning coal again, and this is all how the politics can change the things. But in fact, if we clean, we potentially amplify the effect of CO2. Another one actually was a first test of this kind of large-scale climatic loops. And basically what it says is there are 3 loops. It's a gross primary production, it's a plant production. This depends positively on the way how much diffuse radiation there is. So if we are having aerosols, it's good for plants because there is diffuse radiation penetrates deeper within the canopy. So there is this loop, and this is positively affecting pyogenic volatile organic compound production. This positively affects secondary organic aerosol formation. And these together then lead to higher diffuse radiation. There's a condensation sink here, which means that the aerosols are growing. And here is another link, which is cloud condensation nuclei. This is positively affecting cloud droplet number concentration, so basically clouds. And here is a negative feedback to temperature. And temperature is positively affecting, but due to this negative effect on temperature, it will full cycle will be negatively affected. So it's actually like this sort of loop. CO2 is increasing temperature and increasing productivity. So there are these 2 loops, and then this was the first test actually that it was ever tested. So it's in Finland. I will also talk about this a bit later. They were really one of the best data sets. Like I was showing for this CO2 rise from Mauna Loa, and then now there is productivity data set, which is, so there are very few in the world where are like really old data sets, more than 10 years. Another thing is that most research has focused on constitutive emissions. Constitutive emissions are these emissions that are present all the time, and all those emissions are actually believed to come from constitutive emissions. This is included in the models. And you see these emissions are present in species that have resin ducts. Like you know the smell from conifer needles, for instance. These emissions that come out of the conifer needles. There's a Norway spruce, a Scots pine, but also in some oak species and poplars, there is emissions. You don't feel it actually, the smell, because our nose is not so sensitive. But these emissions are present there all the time, and also in some species there are constitutive emissions. There is no clear understanding why some species are constitutive emissions, why some are not. So there is also research going on to understand this. But basically, some species are, some species are not. In the case of stress emissions, I just wanted to show you also that we have been done quite progress in modeling these emissions, and this is included in different model scenarios, and then it's also a book I edited together with Russ Monson from Arizona University. So this is mainly about constitutive emissions. Now, any stress is actually leading to emissions of also volatiles. It's a different volatiles. Here is an example of C6 aldehydes, which is also called green leaf volatiles. This is the smell of cut grass. If you cut the grass or you crush a leaf, this is the smell you feel. These are the C6 volatiles, green leaf volatiles. So they come from absolutely any stress leads to emissions of these compounds. And later, of course, as gene expression goes on, there is specific monoterpenes, sesquiterpenes, and so on. And this is, and altogether, worldwide, if you look at all different pathways, there is a lot of these compounds are made. And this is a topic of the European Research Council Advanced Grant that is started 1st of May this year. So, where we try to understand what is the role of these compounds in biosphere-atmosphere system. Here's an example of constitutive emissions of Norway spruce. You see this peak of limonene, and then there is Panchavosan monoterpenes. Is a healthy spruce. Limonene is a monoterpene. And the thing is that this is emissions that is included in these big models. This is basically this 1,100 teragram. It's this constitutive emission. But look what happens if this species is attacked by spruce spider mites. you see this bunch of compounds that are emitted. This is like methyl salicylate. Here are sesquiterpenes. Sesquiterpenes are also like in everyday life. Limonene is a compound that is included in all household chemicals. If you look at the label, it's included limonene. But these are included in more like fragrances and also quite common. But you see these major emissions, and then in fact the emissions are increasing 10, 100 times in some cases. And this is not included in any of the model set. That's very important, especially when it's getting warmer or when there will be more stressful environments. Is a big problem. Here's another example for Cabere Busaria, white moth emissions. Here is also, you see, this monoterpene emissions. Here is like a day -1 is non-infected, very low emission, and then it goes up. And in the case of control, it stays low. So it's actually like really emissions. It's a non-emitter under normal conditions. We say non-emitter because it's non-constitutive emitter. In the case of constitutive emitter, it always emits something. But here is a strong emission. Here's another example in the case of rust fungus infection in willow. It's an energy plantation plant, very important in Estonia. And you see it's a constitutive emitter. And then it starts to emit like many sesquiterpenes upon infection. So in fact, being an emitter, it starts to emit much more actually in the conditions of stress. Also, why this is important in the future, there is, people think that it will be much more biologic infections because it will be warmer, the insect generation will be shorter, and also it's many places it will be more humid, which will mean more pathogen fungal attacks. Also, because of the plants contain more sugars at higher CO2, the herbivore stress will be probably larger. And here's an example of this ponderosa pine beetle, mountain pine beetle infection in ponderosa pine in the United States. It can really kill several trees. You see these brownish areas which are all infected trees, and here is like a picture taken from satellite. You see these areas in Canada. It's really large areas can be impacted, and this all converts to these emissions. So this is actually like, during these major attacks, it will be like a emission volcano, like huge emissions. And then this is like, impact will be very big. And then here's another example of abiotic stress. This is heat stress. You see this green leaf volatiles being emitted, cold stress, drought stress. So it's like kind of boring, but you see this kind of also effects that this, the more severe the stress, the stronger the emission response. And we have analyzed actually past data. And here's emission factor, how much the plant can emit. And then we have realized that actually we have really underestimated the contribution of these induced emissions. These are constitutive emissions. And in fact, we have realized now that these emissions here, they are induced because they're dominated by different compounds. And they also respond differently to environment, temperature. These emissions are responsive to light as well. Another example is from a very important northern hemisphere species is birch tree, and here's emissions of monoterpenes and sesquiterpenes, which is all actually induced. If we analyze the emissions under normal conditions, it's very small, and then like in the conditions of stress, it leads to induced emissions. So these are the periods of stress in different times of the year, which are not really included. Also, just to give you an idea what we are doing in reality is right now we started a large screening program to really understand how responsive the plants are, because really to make it quantitatively, we need to understand how much emissions are increasing in response to a given stress. So this is what we are studying in these days. And here's this plant enclosed in plant chamber where all environmental conditions can control. And here's a GC gas chromatograph mass spectrometer. Here's proton transfer reaction mass spectrometer. These are like boxes, and this is what I don't normally tell to students, but just these boxes, if you would like to attach a price label, this is a price of Ferrari or something. It's very, yes? Yeah, I'm actually finishing. It's like, yeah, it's...
Temperate and tropical forests have exactly the same. Are you noticing any difference in this big gush of volatiles you're talking about, like volcanoes coming out from plants? Is it just in the temperate forest or also in the tropical forest?
Oh, it's in both places, yeah. But we actually right now really, the thing is actually that for a given level of environmental conditions, the level of stress plant is feeling is different. So this is actually like a key thing to really attach, make this in a quantitative manner. That say a given temperature in tropics actually, they are much more resistant to heat stress than in the case of temperature forest. And so it's a given rise in temperatures, it depends very much where we are and which pixel in the world. So, well, here is a system, how it works. And then basically we are trying now to develop a model actually. Because these measurements are very good, but we need completely different model than is used for constitutive emissions. And now we need for induced emissions, we are right now developing a model which is starts from stress perception through plant response. And finally, I wanted to finish with this one. I mentioned about the huge technologies that they have in Finland, and is led by Professor Markku Kulmala, who is also co-father of all this business. And what we also, these measurements in forest station, I mentioned they have one of the best data sets in the world, a long-term data set, and this is called Station for Measuring Forest Ecosystem Atmosphere Relations, and we are trying to close this actually. And exactly the same technology to use in Estonia to scale up, because we really need these large-scale fluxes. One thing is to do the work in the lab, but the other is really to say something at the ecosystem scale and scale it up. And then finally it will be scaled up, of course, at world scale. This is supported by a large-scale infrastructure project. We have Estonian infrastructures roadmap, and this is one roadmap object is Estonian Environmental Observatory, which is where we are developing this station. We were, by the way, also the first people in Estonia starting to measure full ecosystem gas exchange fluxes. This is like a routine technology in the world, but it's quite expensive also. So it's like most of these stations are, there are very few in tropical ecosystems, mainly in North America, Europe, and Japan, some in Australia. And then there was nothing in Estonia so far. So we started to measure these, and now we have also got another Ferrari, or even more expensive than Ferrari, is a proton transfer reaction mass spectrometer with time of flight sensor. So this is what we are going to put in the forest. Well, and then here is also like a final picture I wanted to show you. Is a mast we are building. Now it's at 80 meters, but it will be 138 meters. So it's the tallest in the Baltic Sea region. It's the tallest in Finland, actually, in Hytala. So it will be very... And here some first aerosol nucleation measurement. You see really that plants are making aerosols. And then we want to look at all these plant species. So thank you.